All right, today we're going to cover cellular respiration. Now that we have discussed the parts of the cell and we've discussed a little bit about metabolism and a little bit about enzymes, it's an appropriate time to discuss energy and how energy, also known as ATP, gets made in the cell. I will let you know that cellular respiration and photosynthesis are probably at least from what I can gather, the two least liked units in biology. And you'll see why as we go through it. It's a lot of chemistry, a lot of chemical reactions that are occurring. But the comfort that you have is that, at least for this class, it is an open notes type situation because we do move so fast. But like I had said before, if you're moving on to AP Bio, this is probably one of the topics that students tend to struggle with the most. And so I'm going to try to be really thorough with these notes. And a lot of what are in these notes is actually the way I'm going to present them in AP. So that's good news. So why are we talking about cellular respiration? Because life requires energy. Life requires energy. We need energy to grow. We need energy to reproduce. As humans, we need energy for our organs to work, muscles to contract, and all of those things. And an organism grows by the process of cell division. Well, cell division, also known as mitosis, requires ATP. Everything that occurs in a living organism is going to require energy. And so they can get this energy, organisms can get this energy from the sun, from consuming other things, other organisms, or from heat and chemicals released by the earth. So when you look at that, you're like, wait a second, what? Well, we know what gets energy from sun or plants or photosynthetic organisms. There are some bacteria that actually can do photosynthesis, and then there's algae. Algae is a protist, it's not a plant, and it can do photosynthesis. Heterotrophs are going to consume other organisms, so that would be like us. And then there are some that can get energy from heat and chemicals released by the earth. These would be like the bacteria that live in deep sea thermal vents. Those are going to be the ones that actually fall into this category. And we don't talk a lot about them because bacteria that live in deep sea thermal vents, we actually don't interact with them like at all. We don't see them or anything. So that's why we don't really talk much about them in this class. All right, so what are the forms of energy? So energy is going to enter the ecosystem from the sun. Producers like green plants and algae are going to use that energy to generate polysaccharides. Now, when we go through photosynthesis, it's going to make a little bit more sense. The sun is used by photosynthetic organisms to make glucose. Glucose, remember, is that monosaccharide. And remember, glucose is going to link together to form starch, glycogen, and cellulose. And so when the glucose is made by the plant or the algae, it's going to link together to form the polysaccharides, like cellulose and starch. It's going to use sunlight, but also going to use carbon dioxide to do that. And I will go over that in detail in the next lecture. All right, so your consumers are going to ingest and release energy. So consumers are going to eat producers and other consumers. And so when you want to look at the relationship and the flow of energy in an ecosystem, you look at a food web. And the thing that you need to pay really close attention to when looking at a food web are the arrows. The direction that the arrow points refers to the direction that the energy is flowing. And so the way I think of it, the fish is going into the mouth of this hawk or whatever kind of bird that is. It looks like a hawk, right? The little rabbit is going into the mouth of the owl. So that's the direction that energy is flowing. And you have different trophic levels. Trophic levels are things that you would talk about in an ecology unit, and since this is a microbiology class, I'm not going to get into that. But you have your producers, your primary consumers, your secondary consumers, and your tertiary consumers, and all of that good stuff. Right, so Well, I mentioned this word a second ago, but consumers are also referred to as heterotrophs. Heterotrophs are going to eat other organisms. And it's from the Greek word hetero, meaning different, and troph, meaning nutrition. Producers are referred to as autotrophs. The word meaning the word auto meaning self, and troph meaning nutrition. So heterotroph, getting nutrition from another organism, autotroph, getting nutrition from oneself or itself. Okay, so those are just the two vocab words that have to do with trophic levels and the flow of energy through an ecosystem. All right. And so when we discuss how energy is made, how energy is used, the terms potential and kinetic energy come into play. And so all of you have taken physics because we do physics at the freshman level. I'm not going to get a lot into that, but all I really want to say is polysaccharides, proteins, and lipids. When something has a lot of bonds, it's storing a lot of potential energy because those bonds have the potential to break. If you recently took chemistry, or especially if you took AP chemistry, this concept tends to really confuse students because in AP chemistry and chemistry you learn energy is released when bonds are made. Energy is used to break bonds. But in this class, we're talking about the entire, like the totality of all metabolism. So we're talking about bonds breaking, but bonds also being made. But we do say in this class, when a bond breaks, energy is actually released because we're thinking of the whole picture. Because if a bond breaks, another bond is being formed somewhere else, and the energy released from that bond being formed somewhere else is greater than the energy that was used to break the bond to begin with. And I think I probably just confused everyone a lot, but I just want you to know the more bonds something has, the more potential energy it's storing. And in this class, when a bond breaks, we're looking at the net production of energy, and we say energy is produced when you see a bond break in this class. Oh, and the chemistry teachers get so mad at me for that. I'm sorry. That's just a conflict that's there in every school because we're looking at it in a different way. All right, this energy is not useful to the cell unless it can be converted into kinetic energy. So in a controlled manner used to create some kind of cellular motion. And so the way you can think of that is that potential energy is energy stored in bonds. When those bonds break, it causes something to happen. And the thing that happens is the kinetic energy, is the energy being used. So you can think of it this way. Let's say you go to the swimming pool, and I don't know why this picture ended up black. There's just like a big black box right here. I'm sorry about that. Let's say you go to the swimming pool, or you go to a lake or something like that, and there's a really high diving board that you want to jump off of. As you're climbing up the ladder to the top of the diving board, you're gaining potential energy. And then once you jump off of the diving board, that's when there is kinetic energy. And so that's kind of what cellular respiration and photosynthesis are like. Photosynthesis is the building up of sugar molecules, so that's the gaining of potential energy, and then you seek a certain kinetic energy being formed in cellular respiration. And so we're going to go over the steps of cellular respiration so you can kind of see what I'm talking about there. So this energy molecule that's being formed during cellular respiration is called ATP. ATP is also known as adenosine triphosphate. And it's also sometimes called the energy currency of the cell. And this is what it looks like. Let me pull this, try to pull this thing down a little bit. Okay, there, I got it fixed. So you can see that ATP actually has a ribose sugar in it, kind of like RNA. And then it has this adenine. But the most important feature of ATP are the three phosphates. And so these three phosphates have these negative charges. And if you know anything about charges, it's kind of like if you put three balloons next to each other, what are they going to do? They're going to repel each other. And so these phosphate groups are like stuck together really tightly. So it's almost like it's spring loaded. And so something that you know is spring loaded, once you let it go, it pops out, right? And so you can kind of think of ATP as like this. These three phosphates are spring loaded. So when one of those phosphates pops off, energy gets released. And so that's how cells are able to do work, is when phosphates pop off. Well, some way, somehow, we've got to use the glucose. So I'm talking about me as a human, as an animal. I've got to somehow take glucose, eat it, and have it turn into ATP. My muscle cells don't know how to use glucose to contract. They know how to use ATP. So it's kind of like if you travel to a different country and you want to spend money, well, you're going to probably have to, and I don't travel very much. Obviously, this summer, I'm not traveling very much. I was supposed to go to the Galapagos Island. There's actually 3 people in this class right now who are supposed to go with me. Anyways, I'm so bitter about that. But anyway, if you're traveling and you want to spend money, there are some places where you're going to have to trade in your American money for their money and you use their currency. It's the same thing. You eat glucose, but glucose needs to be traded in and made into ATP, and then ATP is the currency that your cells are actually able to use. It's very special and useful because of the potential energy it has, and it can be quickly released. So kind of like what I was saying with the spring-loaded phosphate groups. When a phosphate pops off, it becomes ADP, also known as adenosine diphosphate. So adenosine diphosphate is 2 phosphates together, and that's kind of like a partially charged battery. But when you have 3 phosphates, it's like a fully charged battery. And this ATP right here, so let's say you want your muscle to contract, ATP would pop off a phosphate, and that energy would be used to slide muscle fibers across each other, which causes muscles to contract. And so kind of like I said, the phosphate group can be added, re-added later, kind of like a rechargeable battery. All right, so let's talk a little bit about the relationships between cellular respiration and photosynthesis. So ultimately, the beginning of this cycle occurs right here. If you can see my little cursor, right here with sunlight entering into a photosynthetic organism. Chlorophyll is going to absorb sunlight. Photosynthesis is going to happen. We're going to talk about that process tomorrow. Photosynthesis is going to happen. Glucose gets made. Organic molecules, also known as glucose, gets made. Oxygen is actually released as a waste product in photosynthesis. And so what's very convenient then is the mitochondria uses the sugar made from photosynthesis and uses the oxygen made from photosynthesis to create ATP for cellular work to occur. The mitochondria gives off water and carbon dioxide as a waste. Well, conveniently, photosynthesis uses carbon dioxide and water along with the sunlight to make the organic molecules and oxygen. It's this beautiful cycle. The waste products of photosynthesis, or the products of photosynthesis, are used for cellular respiration, and the products of cellular respiration are then used for photosynthesis. The only thing extra that's coming in is the sunlight. So I just think that's really cool. And so the chloroplast is where photosynthesis is going to occur, and then the mitochondria is where ATP is going to get made for cellular respiration. And so cellular respiration has 3 stages. So what I want for you to know from this picture is there are 3 stages to cellular respiration, and they go in this order from left to right. The first stage is called glycolysis, and it occurs on the outside of the mitochondria in the cytosol, which is like the empty space, or like the jelly-like fluid of the cell. The next step is the Krebs cycle, and then the next step is the electron transport chain. And then what I also want, so I just want you to know, glycolysis occurs outside the mitochondria, Krebs cycle and the electron transport chain occur inside the mitochondria. And then the other thing I want you to know from this picture is the electron transport chain is going to produce the most ATP, the most ATP. So let's talk about each one of these stages of cellular respiration, starting with glycolysis. Glycolysis is the first part of cellular respiration. It occurs in the cytosol outside of the mitochondria, and it literally means the splitting of sugar. So honestly, the only thing that's happening during glycolysis is that glucose is getting split into two. Glucose is getting split into two. And so if you look at this mitochondria right here, this is under a really fancy microscope, the cytosol right here is where glycolysis would occur. In order for glycolysis to begin, though, there's an investment phase. So the only way that cellular respiration can get kicked off or can get started is if 2 ATP are used. And you're like, wait a second, cellular respiration is to make ATP. Why are we using ATP? And what I would tell you is, well, what if you were going to start a business? Your ultimate goal is to make money, right? But the only way to make a really good business is to first spend money. And so you got to spend money to make money. You got to spend a little ATP in the beginning to get the whole process kicked off. Then you end up making a profit of ATP. So there's an investment phase. 2 ATP are needed. And then some other cool things happen. And what you need to know is that glucose ultimately is getting split into 2 pyruvates. I would underline that word pyruvate. Actually, I think you're filling in pyruvate. No, you're not. I need you to underline it. I'm looking at the paper as you're filling it in. All right, so let me just kind of reiterate. Glycolysis is the splitting of glucose. There's an investment phase. You use 2 ATP because you got to spend money to make money, right? And then glucose is getting split into 2 pyruvates. So I would underline pyruvate. The second part of glycolysis actually is going to involve the payoff phase. So you spend 2 ATP, you end up making 4. So then what's your net? Like, what are you making? What's your profit? You're making 2. So if you spend 2, then you make 4. You're ultimately making 2 ATP extra than what you started with. You also have this thing called NAD+. NAD+ is coming and taking hydrogens from that original glucose. All right, and I think this is what is kind of confusing about cellular respiration is it's not just like this linear, I mean, it's kind of like a linear pathway, but there's multiple things happening at one time. Glucose is getting split into 2 pyruvates, but along the way, ATP is being spent, ATP is getting made, NAD+, which is considered this electron shuttle, is coming in and taking hydrogens. Where is NADH going? It's going to this thing called the electron transport chain, all right? And that's ultimately the third phase of cellular respiration where the most energy gets made. So I hope what you can see here is glucose is getting split. You make a net of 2 ATPs. And if you are confused, that's okay. It's normal to feel confused at this point. You can go so deep into glycolysis, it's unreal. I have to show you the picture of it. Sorry, my slide got away from me. But what I ultimately just need you to know is that 1 glucose gets split into pyruvates. Pyruvate can also be called pyruvic acid. That can be kind of confusing. Kind of like citrate can be called citric acid. So pyruvic acid and pyruvate are the same thing, if you want to make note of that. 4 ATP get made, but 2 ATP were used, so you have a net of 2 ATP getting made. And then NAD+, just this random compound. It's kind of like the Uber or the taxi driver of cellular respiration. It comes in and it picks up hydrogens and electrons, and it's ultimately taking them to electron transport chain. So glycolysis is going to produce ATP pretty quickly, which is one of the advantages. Another advantage is that it's anaerobic, meaning it doesn't require any oxygen. So many prokaryotes are going to rely heavily on glycolysis because they don't have a mitochondria. And eukaryotes like yeast are going to use glycolysis where oxygen levels are insufficient. So when you mix yeast, sugar, water, and then you close, you put it in a closed system, it runs out of oxygen, and eventually it's going to undergo anaerobic respiration, also known as glycolysis, but it's going to produce ethanol as a byproduct, and that's then called fermentation. And we'll talk about fermentation just a little bit here. So the process of generating ATP only from glycolysis is called anaerobic fermentation. And so glycolysis is only going to proceed as long as there's enough NAD plus from NADH. This part right here is probably going to feel a little bit confusing, but just so you guys know, the other cell bio classes, they're getting these same notes. And if you're confused, that's okay. What I want you just to know is glycolysis is something that doesn't require oxygen. It's something that prokaryotes will do to make energy, but it's also something that eukaryotes will do to make energy when there's no oxygen. And so when yeast, like I said a minute ago, are undergoing glycolysis where there's no oxygen, they're not going to be able to regenerate NAD plus unless ethanol gets made. So remember, NAD plus has to come in and take the hydrogens off of glucose, and so alcohol gets made as a byproduct. Same thing with lactic acid fermentation. When your muscle cells run out of oxygen, they will then undergo fermentation and will make lactic acid as a byproduct. So really, ultimately, what do you need to know here? You need to know that when eukaryotes run out of oxygen, they revert to doing glycolysis only. But then they also make these byproducts. Yeast will make alcohol, your muscle cells will make lactic acid. And if you're feeling confused, that's totally normal. Totally normal. There's a lot to go over with fermentation, but I think we're just kind of skimming the surface here, and that's okay. For this class, that's going to be okay. Right? So like I said, eukaryotic yeast and some prokaryotes do alcoholic fermentation. And this is what's used to make alcohol beverages, and it's also what causes bread dough to rise. Multicellular organisms, including humans, carry out fermentation using lactic acid fermentation. Muscles aren't receiving sufficient oxygen, and so people will say, oh, it's the lactic acid buildup that causes your muscles to be irritated. Well, actually it is. It's the decrease in pH that irritates muscle fibers. So if you were to do like a wall sit right now, and y'all know what wall sits are. You lean up against the wall and you bend your knees. Eventually it'll start burning. Well, this is why, because your muscle cells are running out of oxygen, and so they revert to doing glycolysis only, and then they're giving lactic acid off as a byproduct, and the decrease in pH, because it's an acid, is going to irritate your muscles and cause the burning. Okay, that was a lot. I mean, that's a lot of information right there. Glucose is getting split. Little bit of energy is made. We talked about some eukaryotes that are only going to do glycolysis when there's no oxygen present and the byproducts. Now let's talk about what happens when pyruvic acid, also known as pyruvate, then leaves glycolysis. So remember, pyruvate or pyruvic acid is made during glycolysis outside of the mitochondria. It is then going to be transported into, so we are talking about eukaryotic cells that have mitochondria that are not undergoing fermentation. There's oxygen available. So that pyruvic acid or pyruvate is going to be transported into the mitochondria, and then it's going to combine with an enzyme called coenzyme A. Coenzyme A helps transport it in. So coenzyme A is an enzyme that helps pyruvic acid, also known as pyruvate. I know I keep saying that, but when you get into, if you are taking next biology AP, I say pyruvate instead of pyruvic acid, but the cell bio teacher this year wanted to use the word pyruvic acid, so I'm going to use that. All right, so coenzyme A is going to help it get into the mitochondria. So here it is, pyruvate. Coenzyme A is helping it come in, and then pyruvate gets converted into acetyl-CoA. Right? Pyruvic acid plus coenzyme A are going to move into the mitochondria. NAD comes in, takes another hydrogen and electron. It's going to move those over to the electron transport chain. Carbon dioxide is released as a waste right here. So that's why when you breathe out carbon dioxide. That's just one of the waste products of breaking down glucose to make ATP. Acetyl-CoA is made. Acetyl-CoA is then going to enter into the mitochondrial matrix, and it is going to combine with an enzyme called oxaloacetate. So acetyl-CoA is going to combine with something called oxaloacetate, all right? And that's happening right here, oxaloacetate. The gist of this entire process is acetyl-CoA and oxaloacetate combine to form citric acid. And then you just follow this in a circle. So what's happening as you're following this in a circle? Carbon dioxide is being released as a waste product. NAD is taking hydrogens and electrons. NAD is taking hydrogens and electrons. A little bit of ATP gets made. FAD, which is another electron shuttle, is going to come in, take more hydrogens and electrons, and then again NAD is coming in, taking hydrogens and electrons. So what is the overall summary of the citric acid cycle? It's that NAD and FAD are coming in and are plucking hydrogens and electrons off of what was originally our glucose molecule. And you don't know yet what's going to happen to those, but I'm going to tell you. So all the citric acid cycle is the taking of hydrogens and electrons away from what was originally our glucose molecule. All right, so for each turn of the cycle, you end up with 1 ATP, 3 NADHs, 1 FADH2. So still not very much ATP getting made. However, a lot of hydrogens and electrons are taken off of that molecule. And on this slide right here, I see an error, and I frequently see this error pop up. I think it's kind of controversial how many NADHs and how many FADH2s are actually made. So if you have 1 ATP, and I'm changing this to, so make sure your paper represents this, 1 ATP, 3 NADHs, 1 FADH2. So if you have 2 turns, you end up with 2 ATP, 6 NADHs, and 2 FADH2. So just make sure those numbers match your paper. And so why are there 2 turns? That's because 1 glucose was split into 2 pyruvates. And the 2 pyruvates come in, and each pyruvate or pyruvic acid is going to take a turn in the citric acid cycle. And this is the end product. And so if you feel like your brain's about to explode, you may want to take a break and just kind of review back over these notes. This is hard. This is a lot of information. Especially in a class where we're so condensed, where we only have 1 day to cover cellular respiration completely. So if all you are walking away with at this point is, what is the end product? Regardless if you really understand what that means, just what is the end product? Where does it occur? Then I think you're good to go. There's just a lot of chemistry that goes into this, and normally when I teach this topic, I do a lot of chemistry beforehand, oxidation, reduction reactions, and stuff like that, but we just didn't have time for it. So it's just one of those things where I'm saying, here's the citric acid cycle, here's where it occurs, here's the outcome. You know what ATP is. You know that CO2 is a waste. I think the most confusion is, what is NADH and what's FADH2 and what makes them so special? Why do we want to make those? Why do we want NAD and FAD to come in and take hydrogens and electrons off of what was our original glucose molecule? Well, I'm about to tell you. This is just a different picture showing you NAD coming in, FAD coming in, NAD coming in, taking electrons and hydrogens off of what was originally our glucose molecule. And so this is the real deal right here, okay? And I put, what questions do you have so far that you want to ask during Zoom? So I think that if you didn't pause before, this would really be a good time to pause and to just kind of look back like, what the heck did we just talk about? And jot those down and make sure that you ask them during the Zoom call. All right, this is the third and final step. Where did all those NAD and FADs, why did we have them? Where did they go? They went to the electron transport chain. The electron transport chain is in the inner membrane of the mitochondria. And this part of cellular respiration is going to use NAD and FAD that were generated during glycolysis and the citric acid cycle. So if you look back at glycolysis, You had some NAD. Then if you look back at the citric acid cycle, and also known as the Krebs cycle, you had some NAD and FAD. They all went over with their little passengers and their passengers. So if you think NAD and FAD of like little Ubers, their passengers are hydrogens and electrons. They go over to the inner mitochondrial membrane where the electron transport chain is located. And so what the electron transport chain is a series of proteins. So if you look at where my cursor is, it's a series of proteins embedded within the inner mitochondrial membrane. And what these proteins do is they pass electrons down. It is like a little chain. They pass electrons down. So why is that so special? So NAD comes in, drops off its passengers, drops off electrons, drops off hydrogens. FAD comes in, drops off electrons, drops off hydrogens. The momentum that is created when electrons are being passed from one protein to another causes hydrogens to be pumped out against their concentration gradient. So I'll say that again. NAD drops off hydrogens and electrons. FAD drops off hydrogens and electrons. Electrons get passed from one protein to another in the electron transport chain. The momentum that is created from passing, because you know electrons are charged, the momentum that is created from passing one electron from one protein to another causes hydrogens to be pumped out against their concentration gradient. So what do you have happening is this accumulation of positively charged hydrogen ions on the outside here. Okay? And so what do these hydrogens desperately want to do? They desperately want to get back in to achieve equilibrium. All right? And so I'll get to that in a minute. When the electrons get to the end of the electron transport chain, they actually join with oxygen along with two hydrogens, and that's where water is made. So this is where oxygen kind of pulls, and it has to do with electronegativity, and once again, I'd have to get into a chemistry lesson, but oxygen is pulling electrons through. They get to the end here, combines with a couple of hydrogens to make water. All right, so now let's talk about the hydrogens. So we talked about the electrons and oxygen and water. These hydrogens are accumulating on the outside here, okay? That creates a reservoir of hydrogen ions in the inner membrane space, and they desperately want to get back across the membrane they were pumped across in order to achieve equilibrium. So they find this door. So they desperately want to get past that membrane. They find this door, and this door is called ATP synthase. ATP synthase is hydrogen's way back in to achieve equilibrium. As hydrogens rush through ATP synthase, a rotor at the bottom of this enzyme turns, and that mechanical energy then phosphorylates ADP to make ATP. And you are like, what is this lady talking about? Like I said, just take what you can from it. We learned about diffusion and osmosis and all of that stuff. Well, here's just an example of an ion diffusing across a membrane with the help of a protein. But what's really cool and unique is as they rush through this protein, this rotor turns. That rotor is mechanical energy, and that mechanical energy is used to phosphorylate ADP, and that's how ATP gets made. And it's a very, very, very efficient process, and that's how so much ATP ends up getting made during cellular respiration. All right? And that's confusing. I mean, this is some high-level stuff. All right? The process of hydrogens going through ATP synthase is called chemiosmosis. Chemiosmosis. So it's kind of like the chemical. It's like the osmosis of an ion almost. All right? And then we can just read back through here. Hydrogen then moves back across the membrane, passing through ATP synthase. The power of this diffusion is used to convert ADP into ATP. This process is called chemiosmosis. All right? High-level stuff. This is crazy. So if you see, I'll go through the process one more time and then I'm going to move on and you just take from it what you will. NAD and FAD drop hydrogens and electrons off at the electron transport chain. Electrons move down the electron transport chain and get passed from one protein to another, eventually combining with oxygen and hydrogen to form water. As electrons are passed from one protein to another, the momentum causes hydrogens to be pumped out against their concentration gradient. Hydrogens desperately want to get back into the inside of the mitochondria, like the inner matrix, and they find a door known as ATP synthase. As they flow through ATP synthase, a rotor is turned and that mechanical energy is used to phosphorylate ADP, making ATP. All right. In the electron transport chain, you have a total of 32 ATPs. And you're like, wait, what? We only made 2 during glycolysis, 2 during the citric acid cycle, and we're making 32 during the electron transport chain. That's why the electron transport chain is so awesome. All right. The net result of cellular respiration, about 40% of the energy in the glucose molecule is transferred to ATP, making about 36 total ATP molecules. The rest is given off as heat or wasted. All right, so those are your total ATP. So you can see why multicellular, eukaryotic, large organisms have to have cellular respiration, because we're so big, we need a lot of energy. All right, so that's the total. Anything that gets, I guess, wasted or left off or isn't used is actually left as heat. All right, and so the question comes up, and we're almost done here. I know this was a lot, but we're almost done. The question comes up, well, what if I'm on an all fat or all protein diet? Well, you're in luck. If you're on a high fat, low carb diet, fatty acids actually will be converted into acetyl-CoA and will enter the citric acid cycle. This is why the ketogenic diet is actually considered the most efficient way to eat, because you completely bypass glycolysis and you enter into citric acid cycle completely. Proteins can be converted into glucose with a process called gluconeogenesis. So proteins can turn into glucose and enter here. Proteins can be converted directly into acetyl-CoA. Proteins can also go straight into the citric acid cycle. So if you had to pick which macromolecule is the best one to eat, you would want to pick protein. You'd survive the longest on it. However, if you eat too much protein, it's really, really hard on your kidneys, so you end up with kidney disease. So it's always a downfall to everything, right? So carbohydrates aren't the only source of energy. Proteins and fats can be used, and they enter into the cellular respiration at different spots. Each is able to do cellular respiration. Last thing that's really, really important is the topic of evolution as it relates to cellular respiration. Glycolysis is really important. Glycolysis occurs in every living organism. All right? The Krebs cycle and the electron transport chain do not occur in every living organism, but glycolysis does. Glycolysis is considered a primitive process. It doesn't require any oxygen. Therefore, the process of glycolysis is used to support the claim or the theory that we all evolved from a common ancestor. And that kind of leads into the endosymbiotic theory, endosymbiosis theory, that a eukaryote-like ancestor engulfed a mitochondria and also engulfed a mitochondria. I don't want to talk about chloroplast. Engulfed a mitochondria, and that's how it became a eukaryotic organism that does all 3 stages of cellular respiration. So I guess what I want you to get from this slide is glycolysis is an ancient process that all organisms do. Therefore, glycolysis is used to prove or to support the theory that we all evolved from one common ancestor. And then the mitochondria is very interesting because it is unlike any other organelle. It is present in all of your cells, and it originated from the egg cell that was provided by your mother. So your mitochondria and your mitochondrial DNA came from your mother. It has its own circular little DNAs like we talked about before, and it's also very, very similar to modern bacteria. And so this is used a lot when we talk about evolution and how mitochondria were maybe once their own little prokaryote-like organisms. All right, that's the end of what I have for you today. Breathe. I do need you to read back over this, and please, please, please come to the Zoom session with your questions. All right, I will talk to you guys later.